Have you ever thought about what happens after death? What if there is a scientific way to cheat death and wake up in the future? Imagine a world where death is not a full stop, but a comma, a brief pause in the narrative of existence. Picture a technology that challenges our traditional understanding of life and death, a concept that blurs the line between science and science fiction. This is the fascinating realm of cryonics. Cryonics is not about bringing the dead back to life. It's about pausing life at the moment of death, preserving the body at low temperatures with the hope that future medical technology might be able to restore that life. Sounds like a plot from a sci-fi movie, right? But what if I told you that this is a reality, a science that is being pursued right now as we speak? Welcome to the intriguing world of cryonics, where death might not be the end. Cryonics isn't a concept from a science fiction novel. It's a real scientific procedure. Let's delve into the fascinating world of cryonics, a technique that lies at the intersection of biology, chemistry, and physics. It's a concept that challenges our understanding of life and death, and it's a testament to human ingenuity and our relentless quest for immortality. Cryonics is the practice of preserving the human body at extremely low temperatures, often at minus 196 degrees Celsius, the boiling point of liquid nitrogen. Why such an extreme temperature, you may wonder? Well, the goal is to halt all biological activity, including the potentially harmful processes that occur after death. It's like hitting the pause button on life, preserving the body in its current state with the hope of reviving it in the future. Imagine, if you will, a future where medical technology has advanced to a point where we can cure currently incurable diseases, repair tissue damage, and even reverse aging. That's the future cryonics is banking on. The preserved bodies are intended to be revived when science has found a way to address the cause of their original death. Preserving the body isn't as simple as just freezing it, though. If it were, we'd all be storing our relatives in the freezer. The process involves replacing the blood with a cryoprotectant solution, a kind of human anti-freeze, to prevent ice crystal formation that could damage the cellular structure. The science of survival through cryonics is a fascinating one. It's a blend of current scientific understanding and a dash of optimism about future advancements. It's a testament to our belief in the power of science and our innate human desire to overcome our biological limitations. In essence, cryonics is a high-stakes bet on future medical technology. It's a gamble that the people of the future will look back at our primitive medical techniques and laugh, just as we do when we think of leeches and bloodletting. It's a wager that our understanding of life, death, and everything in between is set to change dramatically. So how does the cryonics process work? It's not as simple as just freezing a body. Indeed, cryonics is a complex procedure, all hinging on the delicate balance of science and technology. The journey starts at the moment of death, or as close to it as possible. The first step is to stabilize the patient, ensuring that the body's vital functions are maintained. This involves cooling the body, administering medications to protect the brain, and maintaining circulation and oxygenation. But things get really interesting when we dive into the nitty-gritty of cryopreservation. This isn't just about freezing, it's about preventing ice crystal formation, which can damage cellular structures. This is where cryoprotectants come into play. Think of these as anti-freeze for the human body. They replace most of the water within cells and prevent ice crystal formation. Next, the body is cooled to a temperature below minus 130 degrees Celsius. But we don't just slam the body into a deep freeze. The cooling process is gradual, carefully controlled to avoid thermal shock. After reaching the desired temperature, the body is then placed into a long-term storage vessel, known as a diwa. These are large insulated containers filled with liquid nitrogen. In these diwas, the bodies are stored upside down. This peculiar orientation is a safety measure. Should there be a leak, the head, which contains the brain, would be the last part to be affected. Now, it's important to note that the goal of cryonics is not simply to preserve the body, but to preserve the brain and the information within it. It's all about the hope of revival in the future, with all the memories and personality traits intact. Cryonics is a meticulous procedure. an intricate dance of science and precision. It's a testament to human ingenuity, a testament to our desire to conquer death. But it's not without its challenges and controversies, which we'll delve into in the next segment. It's a meticulous procedure, all done with the aim of preserving the minute details of the human brain and body. Cryonics, like any other scientific concept, isn't without its critics. One of the primary criticisms against cryonics is the current impossibility of revival. As much as we'd like to imagine a future where we can freeze ourselves and wake up in a different era, the reality is that we're not there yet. No one has ever been revived after undergoing cryonic preservation. Scientists are still grappling with how to thaw a cryonically preserved body without causing irreparable damage. The formation of ice crystals can rupture cells, leading to a condition known as freezer burn. Not exactly the ice cream sundae of eternity we were hoping for, is it? Secondly, let's talk about the elephant in the room, the cost. Cryonics isn't a budget-friendly option. The price tag for this shot at a second life can range from tens of thousands to over $200,000. This doesn't even include the annual membership fees to cryonic organizations. Some critics argue that resources spent on cryonics could be better used to improve quality of life now, rather than investing in a future that may never come. Lastly, we must consider the ethical implications. If cryonics becomes a reality, who gets to decide who is preserved and who isn't? Are we looking at a future where only the rich can afford immortality? And what about the psychological impact on those left behind? It's hard enough to lose a loved one, but imagine having to grapple with the idea that they might come back to life one day. But let's not forget that every revolutionary idea faced resistance initially. The Wright brothers were ridiculed for their attempts at flight. And now we have planes crisscrossing the globe every minute. Despite the criticisms and challenges, there are those who choose to take the leap of faith. So, what does the future hold for cryonics? As we gaze into the crystal ball of technological advancements, there's a glimmer of hope for cryonics. The next big leap in this field hinges on two key areas: nanotechnology and regenerative medicine. Consider nanotechnology. Imagine tiny, highly specialized robots, or nanobots, capable of repairing cellular damage at a microscopic level. These nanobots could potentially reverse the damage caused by the freezing process in cryonics. They could mend ruptured cell walls, restore damaged DNA, and even clear away harmful substances that accumulate in our bodies over time. Now, let's shift our focus to regenerative medicine. This is the exciting field of science that's all about repairing, replacing, or regenerating human cells, tissues, or organs to restore or establish normal function. The potential here is enormous. We're already seeing breakthroughs like lab-grown organs and 3D printed tissues. It's not entirely outlandish to imagine a future where we could regenerate a whole body using the patient's preserved brain as the starting point. But it's not just about the technology. It's also about the research. Around the globe, scientists are dedicating their lives to unravel the mysteries of cryopreservation, striving to improve techniques, minimize damage, and increase the chances of successful revival. Every day, we're inching closer to turning what was once considered science fiction into reality. However, let's not forget that these are still early days. The road to successful cryonics is fraught with challenges and uncharted territory. It will require not just technological breakthroughs, but also societal acceptance, ethical considerations, and legal frameworks that support this new frontier. In the grand scheme of things, cryonics is more than just a quest for immortality. It's a testament to human ingenuity and our ceaseless pursuit to push the boundaries of what's possible. It's a bold leap into the unknown, a daring defiance of the inevitable. The future is uncertain, but one thing is clear. Cryonics pushes the boundaries of our understanding of life and death. Cryonics is a fascinating intersection of science, philosophy, and the human desire for immortality. It's a topic that takes us on a journey, navigating the intricacies of survival, the complexities of the cryonics procedure, and the multitude of criticisms and challenges that surround this field. At its core, cryonics challenges our understanding of what it means to be dead. It pushes the boundaries of science, daring to question the finality of death and our ability to cheat it. It's a testament to the unyielding human spirit, our quest for knowledge, and our insatiable curiosity. While the future of cryonics is still uncertain, it continues to evolve, with advancements promising to address present-day criticisms and overcome existing challenges. It's a field that is as complex as it is controversial. Whether you see cryonics as a scientific marvel or a fool's errand, it certainly gives us a lot to ponder about the mysteries of life and death.